Okay, everyone, here are the notes for the cell membrane. This goes along with Chapter 5. This is the second set of notes in Chapter 5. And the standards are that you need to be able to predict the movement of water and other molecules across a selectively permeable membrane. So we're going to talk about how that happens. A quick review from Chapter 4. You have plasma membrane, cell membrane, and phospholipid bilayer, for our purposes, all mean the same thing. And the image that they should bring to mind is this phospholipid bilayer. It's made of phospholipids. So here's the glycerol head and the two fatty acid tails. There's a kind of what it would look like if the atoms took up space. If you recall from Chapter 4, we talked about the fluid mosaic model, where these phospholipids are not actually attached to each other in any way. The only thing that's holding the cell membrane together is actually the water pressure from the outside and the inside of the cell. The fatty acid tails do not like water at all. In fact, they're called hydrophobic. And in their quest to seclude themselves from any water molecules on the inside or outside of the cell membrane actually holds together by excluding the water away from these fatty acid tails. And something interesting, if the water pressure increases on the outside of the cell, then the water molecules can actually just sort of slip right between those phospholipids and travel to the other side of the cell. The same thing is true for the inside to the outside. If the water pressure on the inside of the cell increases, those water molecules will slip right through the cell membrane. Now, if the water pressure increases dramatically on the inside of the cell very quickly, then as those water molecules push their way through, they may actually open up a hole, causing the cell membrane to burst or lyse, and the organelles start to come out, the cytoplasm starts to come out, and at that point the cell will die. So we're going to talk a little bit about that today. A quick reminder from Chapter 4. One unique feature of eukaryotic cell membranes is that they have cholesterol embedded in them, and this is something that prokaryotes, like bacteria, do not have. This picture is right out of your book, and if you haven't taken a look at it yet, I would suggest that you do. One thing that on the EOC, they love to put pictures of the cell membrane, and they love to ask you questions about the cell membrane, and this picture does a really good job of summing up a lot of the features that they may ask about. You have your phospholipids. Here's the glycerol head and the two fatty acid tails. You have your cholesterol molecules that I just mentioned, and they're here in blue. You also have enzymes that can be embedded in the cell membrane. We studied those already. The ones that we studied, oftentimes we imagine them as free-floating in the cell or in your stomach acid, but they can also be embedded in the cell membrane. In the last chapter, we talked about carbohydrates and that they're made of simple sugars. So look, even these little things look like they're little hexagons, right? Because carbohydrates make rings with their carbon atoms. So these carbohydrate tags identify the cell, and they're attached to a protein that is embedded in the cell membrane. Now, everything in this picture that's purple is a protein, and these right here are receptor proteins. You're not required to really know much about those for this chapter, but they can identify things outside the cell, and the way they do that is in the last set of notes, you watched the amoeba engulfing a paramecium, and one of the questions that you all have asked is, how did the amoeba know that that was something that it should eat? And one of the ways that it knows are these receptor proteins. As that paramecium bumps into the amoeba, perhaps it's carbohydrate tags or something else on the surface of the paramecium, triggers this receptor protein. And what happens here is it is like a trigger. If you imagine a long chain of dominoes branching out from this receptor protein into further inside the cell, and what happens is when the amoeba brushes up against food, it sets off a chain reaction that says, hey, expand the cell membrane, start to wrap it around this. This is food. Had the amoeba bumped into some other inorganic material that wasn't food, it would not have triggered that chain reaction from that receptor protein. So this is how cells communicate with each other. The last one over here is where we're going to focus for the rest of the notes, these transport proteins. This is how substances move across the cell membrane that cannot just slip right through the phospholipids. And this is vitally important. So we're going to spend the rest of the notes talking about the transport proteins. All right, selectively permeable membrane. Back in middle school, you learned that word and that it meant some things can pass through and some things can't. Well, let's look at the reason why. One of the things that you have to look at is size. If the particle is very small, it can pass through the phospholipids. If it's a very large molecule, then it's not going to be able to push its way or wiggle down through those phospholipids. More important, though, than size has to do with solubility and charge. If the particle is positive or negative, it won't pass through the phospholipids no matter how small it is. The same thing is true for solubility. If the molecule is not soluble in lipids, then it doesn't matter how small it is, it won't pass through the cell membrane. So let's take a look at some examples. If you remember back from middle school, ions are very small charged atoms or molecules. So, would they be able to slip through the membrane, or are they going to need some help? They're going to need some help. What about a sugar molecule, which isn't huge, but it's pretty large? It's going to need some help as well. Gases like oxygen and CO2. These are our cells need oxygen, and they produce carbon dioxide as a waste product. And these gases are actually small enough to slip through the phospholipid bilayer. In fact, the O2 form of oxygen and carbon dioxide, along with water, are some of the only things that can pass through the cell membrane. They are not the only, but they're the only ones that we are going to talk about in biology at this point. So if you can remember that those three can pass through the cell membrane without any help from transport proteins, then you'll be in good shape. So if some things need help to get through the cell membrane, where is it, what does it come from, and how does that happen? And the answer is transport proteins. Sometimes they're called carrier proteins. And they allow molecules to move across the membrane. A distinction you need to make is that some transport proteins do not require any energy. They are passive. Some transport proteins do require energy and they're active. And again, because the test makers like to put pictures of cell membranes on tests, let me show you what a picture of a passive transport protein would look like, and I'll show you a picture of what an active transport protein may look like so that you'll be able to tell the difference. So here's the picture of the passive transport. Take a minute and just pause the video and look at this molecule. It is a step-by-step, 1, 2, 3, 4. Okay. Now, I'm going to make a connection back to our last chapter, almost 2 chapters ago, about enzymes. We know that enzymes were made of proteins. This purple thing here is also a protein, which means the way this functions is our molecule here. Let's just say this is a sugar molecule. And this protein, just like an enzyme, has an active site. So here's something very important that you haven't written down that maybe you should. Transport proteins are specific to one type of molecule. Transport proteins are specific to one type of molecule. Just like enzymes were specific to one type of substrate, carrier proteins are specific to one type of molecule. So in this case, if these yellow spheres are sugar molecules, then as the sugar molecule sits down, and you can think of this as the active site, it causes a change in the shape of the protein. And that change in shape sort of pushes the molecule into the cell. When the molecule is released, the protein goes back to its original shape, much like the enzyme that when the substrate attached It changed shape, which catalyzed the reaction, and when the products were released, the enzyme went back to its original shape and was reusable. This acts in much the same way, except it's doing something different. It's transporting a molecule instead of catalyzing a reaction. This is a picture of an active transport protein, meaning it requires energy. In our last set of notes, we talked about ATP, and in the diagrams on the standardized tests, if it's an active transport protein, meaning it requires energy, you will always see the ATP molecule. It may be labeled like this, or you may just see this little A and 3 little P's for the phosphates here on the back. In this case, what's happening, you have your transport protein here, and even though the sodium molecules, let's say they come in here and look, they're charged, so they're not going to cross the membrane on their own, the sodium molecules are going to attach to this side, but that isn't going to cause the protein to change shape. You actually have to add this phosphate group from an ATP. You get ADP out of the reaction, and the phosphate causes the transport protein to change shape. The sodium molecules are released outside of the cell, and our potassium molecules, which are positive, by the way, that's incorrect, our potassium molecules attach to their sites, but again, this will not change the shape of the protein. When the phosphate is released, the protein goes back to its original shape. So, the dead giveaway between these two transport proteins, one will have ATP involved. It'll be somewhere in the picture, or you'll see a phosphate group attached to it, and you'll see ADP over here, adenosine diphosphate. You'll see something to do with this energy molecule, meaning it requires energy. Passive transport proteins, you will not see that ATP or ADP anywhere in the picture. So if you can do this quick review, you're in good shape.